don't know if this is going to work. You want to record?
Like I know how?
You just have to point it at me.
It's going to be like the Bourne movies, you know?
Well, yeah, it really doesn't come up high enough to. Doesn't have a big enough figure.
Okay. Well, as you all know, I got to talk about bZIP transcription factors. So I thought I'd first talk about what bZIP stands for. It's the basic region leucine zipper. And these transcription factors must contain a bZIP domain. It could be a bZIP1, bZIP2, or an MAF domain. These domains are 40 to 80 amino acids long, and they contain two motifs: a basic region that's involved in DNA binding, and a leucine zipper that's required for dimerization of the transcription factor. I've illustrated a bZIP transcription factor on top here. One of the criteria for this family is it should not possess helix-loop-helix domains, which I've shown right there, so we don't want that. Leucine zippers are put together based on the structure of the alpha helices that compose them. And what they have is a thing called heptad repeats. It's just 7 amino acid repeats with a leucine at the D position. What that means is heptad repeats positions in them are labeled A through G, so leucine falls at the D position. This creates an amphipathic alpha helix, which means it's hydrophobic and hydrophilic. And you can see, like, at every D position, you got a leucine. So this D position kind of spirals around each alpha helix, and it causes the formation of a coil-to-coil. And the hydrophobic region serves as an area for dimerization. The bZIP family is extremely large. It's got something like 78, 79 members in Arabidopsis, 90-something in rice. So very large, highly conserved through across species and stuff. And it's also got a number of functions. So one would ask, how does such a highly conserved protein serve so many different functions? Like, how does it recognize all this different DNA? So what they've done is some mutation analyses to figure out which amino acids near the DNA binding region were the most important. You can see down here that simple changes affect the hydrogen bonds and van der Waals forces that come into contact with the nucleotides. And simple 1 or 2 amino acid changes can give it an entirely new binding sequence. bZIP, like I said, has many functions, from tissue differentiation to seed storage and protein regulation. There's tons and tons of literature on it, and I could finish the semester telling you about bZIPs, but I'm going to stick with AT-bZIP11 for an example today. This one was discovered in 2008, I think. And what was interesting to me about this particular bZIP is it's translationally controlled by sucrose. Now, the way this happens is in the bZIP mRNA, in the 5' UTR, there are 4 upstream open reading frames, or UORFs. And some studies have found URF2 to be the one that's absolutely essential for sucrose-induced repression of translation, or SERT. And this UORF2 encodes a sucrose control peptide. It's about 42 amino acids long. And that's also about the same size as the excitome on the ribosome. So since it's coming in upstream, if you have a bZIP mRNA here, you have 4, 3, 2, 1 UORFs. And then you have a start site. It's about 38 amino acids away. So when it comes along, it picks up the start codon of UORF2, in the presence of sucrose. And the 42 amino acid long SC peptide blocks the exit tunnel when the ribosome gets to the main ORF start site. So it can't initiate translation there. And they've termed this leaky scanning dependent mechanism. And in the study, they also found that SERG is independent of diurnal cycles, hormone signaling, glucose signaling, and starch levels. And they checked all this stuff because sugars and hormone signaling pathways are very intertwined with one another. So they just wanted to know if this thing is independent of all that. So the activation potential is greatly enhanced by SNF1 related kinases, which are involved in the stress response. And if you have extremely high hormone, extremely high sugar levels, you can get a stress response from that. So I think there's something going on there. I haven't put the whole thing together in my head yet, but it's very interesting, I think. ACGT has been identified as the core promoter element for transcriptional activation. In one paper, Smeekins et al., I think, found that it was previously known to be involved in amino acid metabolism, but they recently found that or expanded its function to trehalose, raffinose, and myoinositol metabolism. And another reason I found the bZIP is interesting is because I'm working with a mutant that has 203% trehalose compared to wild type and 187% sucrose. So I thought maybe this guy's involved somewhere. And I also looked at the evolution of the family. It evolved from 4 founder genes. These 4 ancestral groups are composed of 13 subfamilies. And a phylogenetic analysis of 250 bZIPs from rice, black cottonwood, and Arabidopsis showed that there's functional equivalency within each of the subfamilies, which isn't very surprising, but good to know. And out of that, I thought it was a cool little transcription factor to talk about, and that's my brief introduction on it. I'll take questions if you have any.
Thank you, Nick. Yay. So it acts as a repressor or activator?
As a repressor.
Okay, and so if there's functional equivalency within a subfamily, what about between subfamilies or between families?
Between the subfamilies? Between the subfamilies, there is, there's definitely sequence similarity and, conserved structural elements and stuff like that. But, if you look at maybe the G group, that's involved in kind of stress responses, the unfolded protein response, whereas S group is more involved in stuff like metabolic reprogramming.
Okay, so you are getting functional separation between the subfamilies.
Yes.
So then the importance of even looking at phylogeny, if you find a new factor, may give you some indication of at least a place to look.
Yeah, it gives you a starting point to characterize it functionally.
And what do they know about bZIP interactions?
Well, they can form homo or heterodimers. There wasn't a lot mentioned on that in the literature.
In terms of it interacting with other transcription factors, or?
Well, no, they can. I'm talking about bZIP dimerization itself.
Yes, I know.
Okay.
Well,
so you're talking about there's, I'm sure, a plethora, a myriad of things they can interact with.